This is our 7th lecture on bioorganic chemistry, and today we will discuss enzyme inhibitors. The substances which reduce enzyme activity and lower the rate of enzymatic reactions, they are known as enzyme inhibitors. And these compounds they may combine with the certain enzymes, but they never serve as substrates, and they can even block the activity of the enzyme to the extent that whole enzyme becomes inactive. And a wide range of naturally occurring and synthetic compounds, they have the ability to reversibly or irreversibly bind to specific enzymes to alter their activity or even block their activity. So therefore these substances which reduce the enzyme activity, they are studied under two different headings or categories, that is irreversible inhibitors or reversible inhibitors. The irreversible inhibitors are those substances which bind tightly to the active site of the enzyme like this, and they alter the activity of the enzyme which is lost permanently. For example, if this is the active site of the enzyme, then the inhibitor will bind covalently to this active site of the enzyme so that substrate cannot interact with the enzyme, and therefore this will permanently inactivate the enzyme for conversion of this substrate into the product. So that is why these types of inhibitors, they are called as irreversible inhibitors. And most common example of this type of inhibition is found in case of compound diisopropyl fluorophosphate, which is called as DFP. This is an example of irreversible inhibitor, and it inhibits the activity of enzyme acetylcholinesterase, which is a very important enzyme for transmission of nerve impulses. So this is a neurotransmitter enzyme. So how this DFP acts, diisopropyl fluorophosphate, this is the structure of diisopropyl fluorophosphate. So when it interacts with the active site of the enzyme, active site of the enzyme in this case is this serine residue, where hydroxyl functional group is present. So what happens is when DFP interacts with this serine hydroxyl group, then H from here and F from here, they are removed as HF, and this phosphorus oxygen bond is formed covalently, and because of the formation of this covalent bond, now this functional group is not available to the substrate to interact to give the products. So that is why a permanent inactivity of the enzyme is caused because of the presence of this diisopropyl fluorophosphate inhibitor. And once this derivative has been formed, then this enzyme cannot function, or this cannot be an active enzyme for the catalysis. So this becomes inactive for the catalytic reaction for which this enzyme was supposed to do. Then is reversible inhibitors. These substances, they bind less tightly to the enzymes, and in some conditions they can be removed from these enzymes So these are called as reversible inhibitors. And the reversible inhibitors, they can be of three types: competitive inhibitors, non-competitive inhibitors, and uncompetitive inhibitors. So for example, if you see this one here, this type of inhibition is called as competitive inhibition, because here the inhibitor interferes with the active site of the enzyme, and because of this interference of the inhibitor with the active site of, this is the active site of the enzyme. So once this has interfered with the active site of the enzyme, it is not possible for this substrate to interact with the enzyme through its active site. So that is why this type of inhibition is called as competitive inhibitor, because there is a competition between the inhibitor and the substrate to interact with the active site of the enzyme. Then second is non-competitive inhibition. So here what happens is that inhibitor interacts with the enzyme in such a way that it changes the shape of the active site of the enzyme, so that this substrate cannot interact with the active site of the enzyme. So because of the change in shape of the enzyme, the substrate is not able to interact with the enzyme, and this shape change is done by inhibitor. So that is why this is called as non-competitive inhibition. Now competitive inhibitors, these types of inhibitors, they compete with substrate for binding to the active site of the enzyme. So for example, if this is the enzyme and this is the active site of the enzyme, so either the substrate can bind to give us enzyme substrate complex, or the inhibitor can bind to the active site of the enzyme to give us enzyme inhibitor complex. So they are competing with each other, substrate and inhibitor, they are competing with each other to give either enzyme substrate complex or enzyme inhibitor complex. So similarly you can see over here, this is enzyme, E for enzyme, S is substrate, and enzyme and substrate, they are to react to give us an enzyme substrate complex, which will lead to the formation of the products. But once they are competing with each other, then enzyme can also combine with inhibitor to give us enzyme inhibitor complex. And this is a reversible phenomenon, competitive inhibition, and this effect can be reversed or relieved simply by increasing the substrate concentration. So if in this reaction I increase the concentration of this substrate, then formation of products will increase and inhibition will decrease. Similarly, if I decrease the concentration of inhibitor, then also the formation of products will be preferred. So this type of inhibition can be reversed by changing the concentration of the substrate. Then these inhibitors, they generally resemble the normal substrate in three-dimensional structure. So that is because of the similarity in the structure of these inhibitors with the normal substrate. That is why there is a competition between the substrate and the inhibitor, and because of that this ability of the substrate to bind to the enzyme is reduced, because there is a competitor with the same type of three-dimensional structure which can also bind to the active site of the enzyme. The inhibitor molecules, they cannot be attacked by enzyme molecules, and since their active site is occupied, so if the substrate concentration is increased, then the inhibitor molecules, they are displaced from the enzyme and enzyme become functional. So as I have told you, that is why this is a reversible phenomenon. So higher substrate concentration than the normal under normal circumstances is needed to achieve the same type of rate of the reaction. Then some typical examples of competitive inhibition. For example, in case of enzyme succinate dehydrogenase, this enzyme is used for dehydrogenation of succinate to fumarate. So what happens is, one hydrogen from here, one hydrogen from here, they are removed in this carbon-carbon single bond to give us a carbon-carbon double bond. And this succinate dehydrogenase is involved in citric acid cycle, and if in this enzyme in addition to succinate we are having, say, oxaloacetate or oxalate or malonate, then the activity of the enzyme changes to a lower activity. For example, in case of malonate, if you see the structure of the malonate and structure of the succinate, the only difference is here in between these two carboxylic acid groups we are having two methylene groups, whereas in case of malonate we are having only one CH2 group. But malonate, if it is present in the reaction medium, then the active site of the succinate dehydrogenase is competitively taken up by the malonate molecule, and this will inactivate the enzyme succinate dehydrogenase towards conversion of succinate into fumarate. So because of that, the succinate dehydrogenase become inactive, and it prevents for the conversion of succinate into fumarate. But if we increase the concentration of the substrate, and substrate in this case is succinate, so this malonate inhibition can be reversed if we increase the succinate concentration. So this is an example of reversible competitive inhibition. Then another example of competitive inhibition which is reversible in nature is the inhibition of ribulose biphosphate carboxylase enzyme, and this inhibition is done by oxygen molecule. This ribulose biphosphate carboxylase enzyme is very useful during photosynthesis, and the normal substrate for photosynthesis is carbon dioxide for this ribulose biphosphate carboxylase, and even a small concentration of oxygen can inhibit the activity of this enzyme, because this becomes a competitor to the substrate. Substrate is carbon dioxide. So oxygen is incorporated in the active site of this enzyme instead of carbon dioxide, and that is why there is an inhibition in the activity of this enzyme. Then is non-competitive inhibitors. These types of inhibitors, they are not very specific, and they bind at a site on the enzyme other than the active site. So this is the difference. So here these will bind on the enzyme other than the active site, not on the active site of the enzyme. So that is why they are different from competitive inhibitors, and this binding will alter the conformation of the enzyme molecule, so that the reversible inactivation of the active site will occur. So for example, if we are having this enzyme having these two active sites, so if one active site is occupied by the substrate, the other active site can interact with the inhibitor, and we can have this enzyme substrate complex, and then this vacant active site of the enzyme substrate complex can also interact with the inhibitor, and we can have an enzyme substrate inhibitor complex being formed. So enzyme reacts with substrate to give us enzyme substrate complex, which can lead to the formation of products. But this enzyme substrate complex can also interact with the inhibitor, because there is an active site other than the active site where substrate has interacted is also present. So that is why this will lead to the formation of enzyme substrate inhibitor complex like this. Similarly, this enzyme substrate and inhibitor, they can interact together because there is a competition between these two. First, enzyme inhibitor complex is formed, then that enzyme inhibitor complex on the other active site interacts with the, this is enzyme inhibitor complex, then substrate will combine with the enzyme inhibitor complex again to give us enzyme substrate inhibitor complex. So this type of inhibition is called as non-competitive inhibitor, because here the substrate and the inhibitor, they interact at the different sites, different active sites of the enzyme. Then there is uncompetitive inhibition. The uncompetitive inhibitors, they are the compounds which reversibly combine with only enzyme substrate complex. So here the difference is that in the first step of the reaction, enzyme will combine only with the substrate, not with the inhibitor. So inhibitor will not react with the free enzyme. Inhibitor will react only with enzyme substrate complex. So this is the difference between competitive or non-competitive inhibition with the uncompetitive inhibition. So here the first step will be that enzyme will react with the substrate only to give us enzyme substrate complex. Then this enzyme substrate complex will react with the inhibitor to give us enzyme substrate inhibitor complex. So this type of inhibition cannot be overcome by high concentration of the substrate, and increase of substrate concentration increases the degree of inhibition instead of releasing the inhibition, because if we increase the substrate concentration, that means more of enzyme substrate complex will be formed, and if more of enzyme substrate complex is formed, that means more of inhibitor will interact with this complex, and more of enzyme substrate inhibitor complex will be formed. So this is the difference between uncompetitive and competitive inhibitors. Then in terms of kinetics, the competitive, non-competitive and uncompetitive inhibitions, they can be differentiated, and competitive inhibition is most easily recognized by determining the effect of inhibitor concentration on the relationship between substrate concentration and the initial rate. So Michaelis-Menten equation will be used. For example, in this case, when there is no inhibitor, we will get a straight line like this, and as we increase the concentration of inhibition, the kinetics of the reaction will change. Then in case of non-competitive inhibition, the decrease in the value of V maximum, that is maximum velocity of the enzyme, but they have no effect on the Maclaurin constant. That will remain constant, but by changing the concentration of the substrate, we can see when there is substrate only, then this type of straight line is there, and as we increase the concentration of the inhibitor, then the kinetics of the reaction also changes. Then in case of uncompetitive inhibition, the increase in substrate concentration increases the degree of inhibition. You can see over here, first there is no inhibition, then we increase the concentration of the inhibitor, and degree of inhibition also increases as we increase the concentration of the substrate. So Km value of an uninhibited reaction, which implies that substrate is more in concentration, effectively bonds to the enzyme in the presence of inhibitor. So on the basis of these kinetic equations, we can differentiate between competitive, uncompetitive and non-competitive inhibition. Thank you very much.